Thanks very much. Actually, I'd like to broaden the topic a little bit to give you a little more, a broader perspective on low-calorie sweeteners and their effects. First, my disclosures. I am a scientific advisor to the low-calorie sweetener of ILSI and occasional scientific advisor to Ajinomoto, which, among other things, makes aspartame. Okay, so what I'd like to do is, as you can see, some of the studies that have been discussed today have popped into the literature and into the popular press in the last several months. If you don't know the background literature on this, you might think this is scary. And so what I want to do is just briefly show you why most people don't get excited about this that know this literature, because the literature is extensive. And I'll just spend a couple of minutes exposing you to this. Some of you may know it already. Second, I'd like to focus on sweet taste receptors in the intestines, because that is a current topic in research and discussed in the media, having to do with these receptors on enteroendocrine cells stimulating certain incretins that may facilitate body weight gain and obesity. And finally, I'd like to consider briefly the issue of low-calorie sweeteners and obesity. And here, another article that's just popped out by Fowler et al. She had one 6 years ago as well. It's an epidemiologic article, but it impugns diet sodas and, in the early ones, specifically low-calorie sweeteners. And I'd just like to juxtapose the epidemiologic concept with the intervention study concept, because there are several very, very high-quality intervention studies that exist out to 18 months. Many of you may not be aware of them. They go back to the 1970s. And these basically answer the question, ABBA, that's derived, what's the causality, from epidemiologic studies. And you should know there are intervention studies that have carefully investigated if you expose someone to a low-calorie sweetener, do they get fat? Okay. So number one, the background related to safety. I just point this article out to you because it's a fairly intense area, because it's mostly legalities. This article does a very, very nice job in summarizing all of this in a not intensely detailed way that is confusing. If you were a lawyer, it's not confusing. I'm not confusing. This is the least confusing article to me. Rulis and Levitt were longtime employees at the FDA. Rulis was in charge of food additives for a number of years. Levitt was the CIFSAN chief for a number of years. So these guys are well positioned to know this topic. So if you're interested in this, there is the least painful way to learn about it. This includes food additive petitions, as well as the so-called self-GRAS process, which has become more popular these days. And just to give you an idea, so you can see the depth of information that's required in getting a food additive petition, and it's essentially the same for what's called self-GRAS. These are all the studies you submit in a food additive petition: genetic toxicity, animals, short-term metabolic pharmacokinetic studies, short-term tox tests in rats, subchronic tox tests, longer, 90 days, non-rodents, usually non-human primates, reproductive studies, teratology, and so forth, looking for birth defects, one-year tox tests with non-rodents, again, usually non-human primates, lifetime up to 24 months, typically tox and carcinogenicity studies with rodents. That's a lot. And additional data is required following FDA review of all of this. So these documents are rather ponderous. Lots of studies are generally done to generate a document like this. Additional data as required. What would those be? Okay, well, I think Jesse talked about aspartame as having those, I think it was Jesse, the 93 studies for aspartame. And this is a review article, something that some of you might be interested in, Regulatory Pharmacology and Toxicology in 2002. It goes through all those questions that the FDA and the public asked after the aspartame food additive petition was submitted. This is just a 90-page summary of all the published studies. So it's a summary of those studies, and you can see just an enormous amount of data that comprise those 93 studies. So this was no small amount of data that were generated for this, and for sucralose and ASK, et cetera, fewer numbers of studies. The reason for aspartame was 93 studies, was that the FDA decided around 1970 that they were going to review all food additives. And so they contracted with FASEB to set up something called the SCOGS panel, Select Committee on GRAS Substances. GRAS is generally recognized as safe substances. Review all of them. And this process took over 10 years. This process occurred right on the cusp following the diethylstilbestrol and beef scare, the removal of cyclamate as a carcinogen, the removal of saccharin as a carcinogen, which the history of this you may or may not know, but I'm not going to go into this. Saccharin's back, actually cyclamate's back in Europe in beverages for your interest. But aspartame popped into the scene right as this process was going on. And so there was a lot of furor and public interest about this entire process. Aspartame came in. There were no non-nutritive sweeteners, and so this one got a lot was a big focus of attention. So this is part of the reason why all of these studies were generated. Okay, so there is just the skinny on the level of investigation that's done. If you have this level of data, the depth of data, then when an occasional study comes along and you know this literature, you may be less inclined to be concerned about the findings, which come up on a regular basis, by the way. Okay, so sweet taste receptors in the intestines as one focal point. As you know, there are a number of different non-nutritive sweeteners, low-calorie sweeteners. They're tremendously variable in their structure. And you can look at sucrose, which is a modified disaccharide, saccharin, which is actually a coal tar derivative, if anybody knows what coal tar is, from the late 19th century, ACE-K, all relatively small molecules. There's aspartame. It's a dipeptide. It's actually the C-terminus of gastrin. Aspartame was accidentally discovered looking for treatments for ulcer. A very simple dipeptide, rebaudiazide A, which is derived from the stevia plant. It's a diterpene structure, steviol nucleus with some glycosidic linkages, and then small proteins, which are generally in the 50 to 100 amino acid range. They're generally perceived as sweet by humans and other primates, not by other animals. They're not there as sweeteners. They're there as proteins that react to pathogens that attack plants. And so why they're sweet, I have no clue, but turns out they are for humans. They're intensely sweet. So if you look, the intensity of sweetness varies from like 180-fold to 200 for ACE-K, all the way up to 500 to 2,000-fold for Brazan, which is a representative polypeptide sweetener. And most recently, another variation of aspartame called advantame, which just has a vanillin residue hanging off of the nitrogen of aspartate that's 20,000 times sugar than sweetener, so sweeter than sugar. So you practically need molecular forceps to get the amount you need to sweeten your coffee. Very intensely sweet. Okay, so the general feeling was up until the 90s that there's such variety in the compounds that are sweet, and considering also the natural sweeteners, that there must be a number of different taste receptors for sweetness. Towards the end of the 1990s, with some very nice molecular biologic probing, sweet taste receptors were plucked out of taste cells on the tongue, and the sweet receptor turned out to look like this. And it was sweet receptor, not multiple receptors, one sweet taste receptor. It's a heterodimer. It's a G-protein coupled receptor. It has structure, therefore, that is typical of G-protein coupled receptors. The sweeteners, it turns out, bind to different sites on this receptor. So you can see aspartame, glucose, sucrose, sucralose largely bind to the class of receptors called T1 receptor, to the so-called R2 subtype of the T1 receptor in the venous flytrap on the exterior of the cell. There is some evidence that it may also bind to the T1R3 receptor for at least sucralose. The problem with this is that there is another receptor There are three components to this T1 receptor family. So R1 is another one. If you put R1 together with R3, you get the umami receptor, which is an amino acid receptor. If you're a human, you taste MSG. If you're a rodent or less, further down the phylogenetic tree, it's not finely tuned. It's tuned to a number of amino acids. So it's basically an amino acid type receptor. So if you have sweeteners tickling the T1R3 receptor, it will confuse, at least theoretically confuse, the umami receptor or the amino acid receptor. And there are some very interesting studies that show this may actually be the case. So you can see that all of these sweeteners bind to different sites on the sweet taste receptor. I'm not going to go into this. Lactazole is an inhibitor of sweet taste, and it too binds to a site different from the venous flytrap portion. So this is different from the other class that's known to be G-protein coupled taste receptor, the bitter receptors. Here, there are 43, I think at last count, separate members of the T2 community focused on specific structures related to bitter taste. If you go to the G-protein coupled receptors related to smell, you find hundreds of receptors, if not more, fine-tuned to different odorants. So it is somewhat of a surprise that you have a single receptor for multiple sweet-tasting substances. Perhaps this means that it's not that when you experience sweet taste, that's generally a good thing in terms of no harm to your body. You don't need more than one receptor. That's a guess. Okay, so once you know what this receptor looks like, then you can go look for it elsewhere. You can make antibodies, go see where they are outside of the mouth, and one of the first places that folks went to was the rest of the alimentary canal. A sweet taste receptor turned up in enteroendocrine cells, EE there, on microvilli, on villi in the small intestine. The dark cells, there are very few EE cells that are interspersed amongst the enterocytes. The enteroendocrine cells do what the name implies. Molecules, nutrients interact with these cells, induce the release of small peptides, hormones. In the case of sweet taste receptor and these EE cells, incretins, which would include glucagon-like peptide 1 and 2, and GIP. In relation to the discussion today, it's mostly GLP-1 that's of interest here. So how would you do a study to know that the enteroendocrine cell actually uses the sweet taste receptor to induce the release of GLP, 1 or 2 for that matter? Well, you could throw a sweetener at it, like a natural sweetener, like sucrose or glucose, fructose, what have you, and you would see a release of the hormones. And then the question would be, is it the sweet taste molecule interacting with the receptor, or is it the metabolic consequences of those cells absorbing the sweetener, in the case of a natural one, glucose or fructose, and metabolizing it, generating ATP and other molecules that certainly could function as intracellular signaling molecules independent of the sweet taste receptor? Well, a way to answer that question is to try throwing sucralose, which looks the closest to a disaccharide and is not metabolized by these cells, throw that onto the cells in vitro. And if there was a response, it would have to be due to interacting with a sweet taste receptor. That was done, and that's what was found. The GLP-1 and 2 were released in response to sucralose. It could be blocked by lactazole, which is the inhibitor of the sweet taste receptor. So, boom, those sweet taste receptors are important in the release of these incretin molecules. Well, a correlation or a suggestion from that is, oh wow, sucralose stimulates the release of incretins. Can that screw up your regulation of carbohydrate flux from the gut, ultimately to the cells that are going to take it up? And as near as I can, sorry, this is just a model that I just described, I won't go into it. And so here's sort of my take on what is being proposed here. This is a hypothesis. However, it's been tested now. So why are incretins linked to low-calorie sweeteners and obesity? So GLP-1 increases insulin secretion. It's an enhancer of insulin secretion. It's the definition of an incretin. GLP-2 acts as a paracrine agent on the enterocytes to stimulate glucose transport through the SGLT1 receptor for glucose transporter. And so you could make the hypothesis that as such. So when you ingest a caloric sugar containing sugar, you increase GLP-1 and 2 secretion by interaction of the sweetener with the sweet taste receptor on these cells to facilitate glucose transfer from the gut into distal tissues. If, however, it's a low-calorie sweetener, and the question here is, it's a hypothesis, does it in fact lower blood glucose because in the fasting state there is no glucose or sugar coming in from the intestines, and if the incretin has their effects, it would tend to decrease blood glucose and increase hunger, and that would an increase in food ingestion and weight gain. If you were eating it with food, you would get excess sugar absorption as a result of the incretin secretions and uptake into tissues, and therefore enhance the laying down of excess nutrients into fat cells and et cetera. So that would be the hypothesis that actually jumps out of the speculations that have been made from some of these very nice in vitro articles. Is that true? Well, I just pick out two examples. One is in the case of fasting, and they're human examples. This is Ma et al. In the American Journal of Physiology in 2009, looking at fasting humans, giving them either sucrose or sucralose at a concentration that you would get in a beverage or at 10 times that concentration, 0.4 millimolar or 4 millimolar. And the open squares are what the glucose response, insulin response, and GLP-1 response are to sugar, to the sugar molecule, sucrose, and you can see nice glucose tolerance-like responses for glucose and insulin. And you see the white box at the top there with GLP-1, a robust secretion of GLP-1 picked up in the peripheral circulation. When you look at the sucralose responses, you can see the black dots and squares, no glucose response, no insulin response, and non-statistically significant responses to either the 0.4 or the 4 millimolar concentration of sucralose. The conclusion being that in a fasting state, that you don't see the predicted responses that would occur if that hypothesis were correct with regard to any of the involved downstream effectors, glucose, insulin, or GLP-1. So this doesn't support the hypothesis. Now, there are a lot of articles out there, human studies, there are 10 or 12 now. They're not all exactly the same, but I think on balance, they tend not to support the idea that there is an incretin-mediated enhancement of nutrient flow into peripheral tissues. So here's the fasting case. Here's an example, and the model is you pre-treat with a particular agent, sweetener or a non-nutritive sweetener, and then you do a glucose tolerance test. You take a load of glucose, and then you see if that modifies glucose tolerance. And so in this case, this is Wu et al. In Diabetes Care in 2013. It's water versus sucralose or ACE-K, or the two together. And that's because a lot of the diet beverages that are around are a blend of sucralose and ACE-K. And these are in concentration appropriate levels for taste. And so you can see for the blood, the 2 times, by the way, minus 100 and 0, this is the time period before the glucose is administered when you've had the water or the artificial sweeteners, and you see there are no effects by themselves, consistent with the last slide. And then you see the glucose response, they all overlap. The insulin responses overlap and the GLP-1 responses overlap, meaning that what you see is the response to the glucose load, and there is no enhancing effect of sucralose or ACE-K or the combination. Okay, so what my takeaway message from that is, and certainly others who've done this that may be in the audience can raise issues about this, but my takeaway message is that the idea that there might be a low-calorie sweetener-induced enhancement of the incretin response doesn't seem to hold up when you do human studies. And finally, low-calorie sweeteners and obesity. And here, what we're looking at is epidemiology versus intervention studies. And so this is Fowler's first study. It's the San Antonio Heart Study. It's just collecting self-reported food intake data, including sweeteners and non-caloric sweeteners, and then correlating the data that were collected with likely incidence of obesity. And here from an epidemiologic study, at least to me, the non-epidemiologist, I would go, fine, you make a correlation, and the correlation and if it's a good one, allows you to say, oh, okay, so there is a correlation between the intake of diet soda, which is really what she was studying, and body mass index, and that could tell you one of two things. Either you drank some diet soda and it made you get fat, or you were gaining weight and you go, my God, I need to do something to control my weight gain, so you started using low-calorie sweeteners. So which is it? She seems to imply these findings raise a question about whether artificial sweeteners might be fueling, rather than fighting, our escalating obesity epidemic, but I don't think that's appropriate given that this is a correlational epidemiologic type study. Well, the question could be answered. That's 2008. Are there any intervention studies that would allow you to evaluate that hypothesis? Yes, there were at that time. I've picked out the ones that I would feel most comfortable defending on a platform. There are many more than this, but these are the ones I like the best. There's from Ted Van Italy's group at Columbia, Pericos and Pisanier, a review article in 1984 that describes two studies that were done. The first one published in AJCN in the late 70s, a 12-day study. It was in a CRC. They were all locked in there, so the variables were fairly well controlled. There are a few problems with this, if anybody remembers the studies, but it showed absolutely no increase in body weight when you switched from normal diet to caloric dilution, in this case with aspartame, because that was the one that was around at the time. Tordoff and Oliva in 1990. This was a beverage study, 3-week study, very good. It was not in a locked center. The Perico study was the only one that was. But just introducing low-calorie sweeteners in the form of diet sodas into a group of subjects in their natural diet and just drink these, eat your normal diet, but by the way, drink these too. I'm not going to spend much time with this. Raben, which is a 10-week study in 2002, a very, very nice study showing, again, no increase in body weight when someone is exposed to a substantial amount of low-calorie sweeteners purposefully introduced into the diet. And finally, the most recent DeRuyter study, which is 18, not weeks, but months in kids. And this is the one I'd like to say a few things about. I love this study. I think it is the best designed study yet done. And so let me spend a minute or two on that. So here it is, if for those of you who haven't seen it and might like to. It's in the New England Journal in 2012. It's a Dutch group. I can't imagine how much money this study cost, but it's done in school children. They recruited 641. It's very interesting. It was done in a school environment where the kids have a morning snack every day, and the snack includes a beverage and a light cake or some such thing, cookie, whatever. Of constant number of calories. But what they did was they took kids who only drank sugar-sweetened beverages. So they didn't take kids who didn't, say they drank water or non-sweetened beverages or low-calorie sweetened beverage. They only used kids that drank calorically sweetened beverages. The reason they said was moral, but actually it introduced a very nice design feature into their studies, and that is it didn't add a calorically dense sweetener to half the group. It took kids that were already taking them and then took half of those and removed the caloric sweetened beverage from their snack. And that's different from the previous studies. So half were given the normal sugar-sweetened beverage and half were given low-calorie sweetened. It was a double-blind study. Every day when they had their snack, 5 days a week, the teacher monitored their intake of the snacks and the beverages, so it had good control of intake. Over the weekend, they took them home, and their parents were reminded to give their kids these beverages over the weekend. And they looked at various anthropometric metrics at 6, 12, and 18 months. I'm not going to talk about more than this. This is the top figure is the most interesting to me, at least the most easily interpretable. This is looking at BMI Z-score in these kids. BMI Z-score, in case you don't know, it's essentially looking at a score that is a deviation from the mean of BMI for age, for sex, okay? So, for example, when you look at the 0 time point, the BMI Z-score is close to 0. In the black squares, which is the kids drinking the caloric sweetener, at 6, 12, and 18 months, it goes up, which means that those kids at 6, 12, and 18 months were deviating positively from the mean BMI score for age in the sex. Okay? And if you look at the open circles, it's not really changing at all. And that's the group that got the low-calorie sweetened beverage over that period of time. So to me, what that says is that the kids who are getting the low-calorie sweetener are not changing in relation to their population at the given age in their BMI, where the kids who are continuing to drink the calorically sweetened beverage are increasing. They're getting up into the higher levels, not incredibly high, but higher, statistically higher BMI range, age and sex adjusted. Very interesting study. They did a lot of things. They measured the sucralose content. It was sucralose ACE-K blend. They were using the sweeteners. They could measure urinary sucralose. They did that over the period of time of this study, and so they could nicely monitor. Their pharmacokinetics were good. There are a few suggestions I would make, but they're minor. So they knew these kids were taking the beverages. They also did a satiety study. There are 3 studies actually published, not just the one, showing that there's no difference in satiety after the ingestion of the low-calorie sweetened beverages. Most of the 93 studies on aspartame were engendered by studies like that suggested that low-calorie sweeteners would immediately stimulate appetite and cause you to eat more food. That turned out to be not true, and it's certainly not true here as well. So satiety, appetite is not affected, in this kind of study. Okay, so let me just summarize the points that I'd like to make. There are large databases on each of the low-calorie sweeteners based on the submission of food additive petitions and self-GRAS petitions. And so when you see new studies and you know these databases, they really cover most of the issues, even the new ones that arise. The low-calorie sweeteners don't release glucagon-like peptides or alter intestinal glucose transport. I think that's a fair statement. This mechanism is therefore not a cause of obesity, in my opinion, although this is still an actively debated topic, I might add. And finally, the intervention trials. They really show you that low-calorie sweeteners do not lead to weight gain. And I like to say weight gain, as Jesse pointed out, there are some data showing that there's actually a weight reduction, but the argument is that they cause weight gain. These studies incontrovertibly show they do not cause weight gain. And so with regard to the epidemiologic studies, this implies that the causality link is likely to be you're getting heavier, and therefore you use these things to try and control your caloric intake. So finally, future research, and that's what my topic was supposed to be. The work with research on sweet taste receptors is really exciting, and it's only been going for about a decade, and there's new and really interesting stuff going on every day. And my feeling is if I were saying where to spend your dollar, I would definitely spend lots of dollars on the basic research in these receptors, taste receptors. Really interesting stuff. Safety studies for new low-calorie sweeteners, obviously, if you're going to go and file a petition, food additive petition or self-GRAS, you're required to collect a lot of new data that basically have to do with safety. Fine, you should. In fact, it's my opinion. I think DeShay is a mistake. I think anything that gets out there that's a dietary supplement that says it's sweet, I think they ought to be reviewed in terms of their safety, but generally they're not because Dietary Supplement Health and Education Act prevents the FDA from requiring that. The older LCS, low-calorie sweeteners, obviously should be looked at in terms of safety as issues arise, as they obviously do. Safety now means not teratology, carcinogenicity, et cetera, but also include things like getting fat, which leads to comorbidities, which are not good. So I'll stop there. Thanks very much.